Hello, my name is Spencer Wozniak, and today we will be looking at artificial intelligence, specifically machine learning in medical microbiology. And before we look at examples in medicine, I'm going to try and introduce some background about machine learning that will hopefully provide an understanding for how it generally works that will help you understand the examples down the line. Before we get into the details, let us first look at some cool state-of-the-art examples of machine learning. So one of the most amazing examples of machine learning is DALL-E 2. This is able to create images based on text input, and shown on screen are just some examples of novel images that were created by DALL-E 2. Another amazing example of artificial intelligence is GPT-3. GPT-3 is an AI that can interpret language and produce human-like text. Shown on screen is just one example of something GPT-3 can do. It is told to summarize a relatively complex passage in a way that a second grade student would be able to understand. GPT-3 has also been shown to perform better than the average human on the SAT, and it's actually capable of writing code that does what you tell it to do. And in my opinion, the most remarkable accomplishment of GPT-3 is its ability to create new AI, actually. An example of artificial intelligence used widely in the scientific community is AlphaFold. Proteins, as you know, are made up of a linear sequence of amino acids that are able to fold into complex shapes in solution. Given a sequence of amino acids, also known as its primary structure, AlphaFold is able to accurately predict what the three-dimensional folded structure would be, which is known as the tertiary structure. Aside from this, it is able to predict novel sequences of amino acids that are able to fold into similar structures as a given 3D protein. So how is it possible that a computer is able to perform these complex tasks? The answer is machine learning. Machine learning is a type of artificial intelligence that is able to learn based on large data sets. Modern machine learning algorithms utilize neural networks, which are based on the way the brain works. Ultimately, your brain is nothing more than a complex neural network. And a neural network is able to learn via a process known as optimization, which tunes weights and biases between neurons in a neural network. So the details on the last slide might seem confusing if you've never heard any of these terms before. So I'm going to try and break it down. So what you're seeing here is an extremely simple neural network in which each circle represents a neuron. The arrows indicate the flow of information, and you can think of them like synapses between neurons in your brain. To see how information flows within a network, we will start by looking at one connection between two neurons. Each neuron is connected to another neuron, and the connection between the two has a weight associated with it. Each neuron has a numerical value associated with it. In this example, the first neuron, the red one labeled N1, has a value of 4. This neuron communicates with the next neuron, N2, via a weight. And so the value of the second neuron is simply the weight of the weight multiplied by the value of the first neuron. So here we have 4 times 0.25, which would give 1 in the second neuron. Now, each connection also has a bias associated with it. Here, we can see an example of neuron-neuron communication with a weight and a bias. Like in the last example, we multiply the value of the first neuron by the weight, so 4 times 0.25 is 1, but here we also add the bias, so 1 plus 2 gives us 3, which is the output neuron, N2. And the notation here may look complicated, but it's really very simple, and might just take a second to look at. Now, looking at the neural network, notice that neurons are not just connected one by one. Each neuron has multiple connections that run into it. Therefore, we need a way to integrate all of the messages coming from incoming neurons. There are many ways in which this can be done, but one of the simplest ways is just to add all of the messages from incoming neurons. So here is an example of multiple inputs to a neuron. Each of the red neurons sends a message to the next neuron. And like we discussed before, we simply multiply the value of each incoming neuron by the corresponding weight, then we add the corresponding bias, and that is the message. And here we get these following messages, and if we add all of the messages from the incoming neurons, then we get the value of the output neuron, which would be 6 in this case. Now, the past few slides explained how a neural network works, but it's most important to understand what a neural network and what machine learning models actually do. So a neural network or a machine learning model is effectively a function that takes in an input and returns an output. The input can be anything you would like: text, numbers, photos, or anything else you can think of can be embedded in a numerical form such that the neural network or machine learning model will understand what it is dealing with. Like I said before, your brain is a neural network. The input to the neural network of your brain includes all of the external and internal stimuli that are detected by receptors of your body. The output of a neural network can be anything from adding numbers to solving a complex problem to identifying patterns in data. And the output of the neural network of your brain is extremely complex and includes things like controlling muscles, sensory perception, managing homeostasis, like managing your blood pressure, heart rate, temperature, decision making, problem solving, emotions, social behavior, and many more. So here's the neural network again. Notice that there is an input on the left and an output on the right. Everything that happens between is the neural network or the machine learning model, which is simply just a function, which is denoted f here. And here we can assign the values a, b, and c to the input neurons and assign the value y to the output. And so here we can see that the neural network takes in the values a, b, and c and returns the value y. And instead of using the letter f, neural networks are typically signified with the Greek letter phi or phi. So going forward, that's the notation that we will be using as well. Now, something you could do with a neural network is predict whether someone is sick based on their heart rate, breathing rate, and body temperature. In this case, A is heart rate, B is breathing rate, and C is body temperature. These values are fed into the neural network, which returns the output Y, which in this case is either a 0, indicating the person is healthy, or a 1, indicating the person is sick. Something else you could do is predict whether someone has an infection based on C-reactive protein levels, procalcitonin levels, and white blood cell count. In this example, A represents the serum C-reactive protein level, B represents the serum procalcitonin level, and C represents the white blood cell count. Again, these three values are fed into the network, which returns either a 0, indicating no infection, or a 1, indicating an infection. Now, looking back to the neural network, if we were to input the average values in a healthy person, as seen here on the left, we would expect to see a 0 on the right as the output, indicating that the person is healthy and does not have an infection. Now, the outputs of the last two examples were pretty simple, and you would likely not need a neural network to solve these problems. However, the output of the network can be adapted to much more complex problems. Now, here is our neural network again, just with one output. Instead of having one output, our network could have three outputs instead. Notice that A, B, and C are still the inputs, but now we have three outputs, X, Y, and Z. So, rather than simply predicting whether someone has an infection, you may want to use a neural network to predict the probability of different types of infections based on these three levels. Here, A, B, and C still correspond to C-reactive protein levels, procalcitonin levels, and white blood cell count, respectively. But now we have three outputs, and X, one of the outputs, is the probability of a bacterial infection, Y is the probability of a viral infection, and Z is the probability of a fungal infection. More specifically, you may even want to predict the probability of different types of bacterial infections based on these inputs. For example, you may want to predict the probability that someone has a gram-positive infection, gram-negative infection, or mycobacterial infection, which would then allow you to give empiric antibiotic therapy based on this. And you could even try scaling up the output to include probabilities of hundreds of specific infections caused by different pathogens. Now, neural networks are extremely adaptable, and the inputs and outputs could be anything you would like. One way to design a neural network or machine learning model is to think of something you would like to predict, like the probabilities of different types of infections. This will be the output of the model. To choose an input, you should think of things that could be useful in the prediction of the output. I used C-reactive protein level, procalcitonin level, and white blood cell count as inputs in the previous examples because these values could be useful in the prediction of infection. And now it's important to note that while inputs and outputs can be anything you would like, you need to realize that the model is only going to be useful if it can learn something about the input that is relevant in determining the output. For example, a neural network would easily be able to recognize that elevated C-reactive protein levels, procalcitonin levels, and or white blood cells means that there is an infection, but it might not be able to distinguish what type of infection based on these values alone. However, if you add data from a differential white blood cell count, for example, the model might be able to learn that elevated neutrophils is associated with bacterial infection, elevated lymphocytes is associated with viral infection, elevated monocytes is associated with fungal infection, and etc. Now, how is it able to learn? Modern neural networks have millions to billions of connections, all of which have weights and biases associated with them, as we discussed earlier. And here we can see a quick comparison between the number of connections that these modern neural networks have compared to the brain, which is much more complex, and that's why the brain is able to perform much more complex tasks than these neural networks we currently have. And basically, it's the operations associated with these connections that allows neural networks to solve extremely complex problems, including finding patterns in data that humans would never be able to recognize, and accurately predicting outcomes based on inputs. Neural networks are trained on large sets of data, which contain thousands to millions of data points. And each data point within the data set has input values and known output values that are called labels. Now, during training, the neural network makes a prediction using its current configuration of weights and biases. This prediction is compared to the label by something known as the optimizer, which can tweak the weights and biases of the neural network. Now again, this terminology might seem very confusing, so we're going to try and illustrate how this actually works. So we're going to look back to this example that was shown previously in which A, B, and C correspond to these three levels, and the output could either be a 0, indicating no infection, or a 1, indicating infection. Now notice that this ABC shape here is the data point, is one data point, and when you run it through the model, you get an output either a 0 or a 1. Now we're going to start with the data set here. Each individual data point, which you can think of as an individual patient, is represented by one of the red columns. Now recall that A is CRP level, B is PCT level, and C is the WBCs. That's what each of these values for the input correspond to. Now underneath, we have labels that indicate whether each data point represents either a sick person, indicated by a 1, or a healthy person, indicated by a 0. Each data point is fed into the neural network, which outputs a value, either a 0 or a 1, based on its current configuration of weights and biases. And we can see that the predictions here are not perfect because the weights and biases have not been changed yet. So this output is compared to the labels by the optimizer. And based on this comparison, the optimizer changes the weights and biases within the neural network in an attempt to cause the neural network to make a better prediction. And here, if we look at the output, we can see that with its new set of weights and biases, the neural network can make a new prediction, which is compared to the labels again, and then the weights and biases are changed again. And we get another prediction, and this cycle repeats itself until the predictions of the neural network are as good as possible. And eventually we see that the output matches the labels, and we would refer to this as a fully trained model here. Now, after training, we also want to verify the performance of the neural network with an independent data set. This data set, known as the validation data set, contains data that the neural network was not trained on. If the model can make good predictions on this data set, it indicates that the model learned something about the input that was relevant in determining the output. And it is important to note that all performance metrics associated with a machine learning model are obtained using the validation data. So in examples to come, values for sensitivity, specificity, accuracy, and error are all associated with model performance on the validation data set. This ensures that the model actually learned something and did not simply memorize all of the data in the training set. Now to the right, we can see a graph of the RMSD, which is just an error metric, versus the cycle of training. A higher RMSD indicates worse predictions, while a lower RMSD indicates better predictions. And as the model is trained, the RMSD on both the training and validation data set should decrease. If the RMSD on the training set decreases, but the RMSD on the validation data set does not, this indicates that the model is not actually learning anything about the input that is relevant in determining the output. Rather, the model is basically trying to memorize all of the data in the training data set. Now that we have a little bit of background on how machine learning works, we're going to look at some reasons for why machine learning should be integrated into modern medicine, as well as a few examples of it actually working in medicine. In the context of infectious diseases, modern medicine utilizes a wide range of technology and equipment to guide empiric therapy. When a patient with an infectious disease comes into a clinic, treatment may begin in the absence of complete information. The healthcare team gathers a patient history and can begin ordering tests to acquire more complete information about the patient. Information about infectious diseases can be obtained via various methods, including culture, nucleic acid amplification, serology, and MALDI-TOF. So, state-of-the-art methods for diagnosing infectious diseases can prove quite costly. Traditional methods that involve culture cost an average of about $140,000 per year per laboratory. A MALDI-TOF machine can cost up to $270,000 up front and costs about $70,000 to run each year per lab. A BioFire panel, which is used to run multiplex PCR assays, costs about $35,000 up front and costs about $214 per test. And serology, which is often the cheapest option, costs about $2,000 up front if you purchase an analyzer, and it costs about $34 per test. Now, compared to the cost of other methods, integrating machine learning into the healthcare setting would not impose a significant cost. All you need to run a machine learning algorithm is a computer, and hospitals would likely not need to spend much on computers as there are already a lot of them in a hospital. Aside from the cost of equipment needed, any additional cost depends entirely on the input of the machine learning algorithm used. For example, if a model requires information from a CBC as input, there would be a $50 cost associated with its use because the CBC costs about $50. However, tests like this are already routinely ordered, so there would not likely be much of an increase in cost if we can use information like this that is readily available. And overall, the low cost associated with incorporating machine learning into the medical setting means that it is highly accessible. So low to middle income countries, as well as underfunded hospitals and laboratories, could benefit greatly from the incorporation of cheap, highly accurate machine learning technology. So let us now take a look at an example of how machine learning could be integrated into medicine to aid empiric therapy. So infants are particularly susceptible to serious bacterial infections, including those that affect the central nervous system. Infants with fevers are commonly brought into the hospital, but many of them do not have CNS infections. However, current methods to predict CNS infections based on the patient presentation are not very specific, leading to many false positives. Once a child is suspected to be positive for a CNS infection, a lumbar puncture is typically performed so that the cerebrospinal fluid can be analyzed. These false positives ultimately lead to unnecessary antibiotic use, lumbar punctures, hospitalizations, and parental anxiety. In a paper titled "Machine Learning to Predict Serious Bacterial Infections in Young Febrile Infants," researchers set out to develop a machine learning algorithm that had a higher specificity than current methods for detecting CNS infections without performing the routine lumbar puncture. They used the same data that would be analyzed according to traditional guidelines as input to their model. This includes data obtained from urinalysis, a complete blood count, a differential white blood cell count, and a procalcitonin test. And these were used as inputs to the model, with the output indicating the risk that a given febrile infant does have a serious bacterial infection. So using data from 1,470 patients, the researchers were able to train a model that had better specificity and sensitivity than traditional methods of diagnosis that don't involve a lumbar puncture. Researchers noted that the increased specificity could have prevented 849 lumbar punctures that were performed on false positive patients. Furthermore, these results could be obtained just as quickly, if not quicker, than the results of traditional methods. Overall, the researchers developed a machine learning model that looks at the same data we have already been looking at and has much better accuracy. It should also be noted that the sensitivity of this model is very impressive and is even higher than the sensitivity of cerebrospinal fluid culture, which does require a lumbar puncture. The impressive accuracy of machine learning models has already been demonstrated in countless areas of science. However, as our computers become more powerful, we will be capable of creating better models, which should only lead to better accuracy. On top of this, we can improve the accuracy of our models by providing the most useful input possible, which we may not even know yet. We will also continue to improve the accuracy of our models by improving the structure of the models themselves. So overall, the field of machine learning is rapidly expanding at this moment, and it may be too soon to say how accurate these models may become in the future. Now, what makes machine learning so useful is that it can be applied to virtually anything, from broad generalizations to specific classifications in almost any field you can think of. This flexibility is what makes machine learning one of the most promising technologies that we currently have. Furthermore, a machine learning algorithm can be as complex as you need it to be, and you can even run multiple machine learning models together to solve a very complex problem. Now, before we move on, let's briefly look at some other current examples of machine learning being applied to medicine and microbiology. Machine learning has been shown to improve the accuracy of secondary bacterial infection diagnosis in the context of COVID-19. Like the previous example, information that is already used to guide empiric therapy was used as input to the model, which included information about patient demographics, complete blood count results, and any clinically significant microbiology data. Now again, all of this is embedded in a numerical form such that the model can understand it, and this model was used to support the diagnosis of secondary bacterial infections when somebody had a primary COVID-19 infection. And it ended up showing a sensitivity of about 80% and a specificity of about 90%. Machine learning has also been applied to guide empiric therapy in the context of Candida auris, which is an emerging yeast that is often resistant to all three classes of antifungal drugs. The input in this model was the sequence genome of a given strain of Candida auris, which not only allowed the prediction of resistance to the three classes of antifungal drugs, but also predicted specific genetic mutations that could be associated with resistance to specific drugs. The researchers concluded that machine learning classifiers are a useful and cost-effective way to identify fungal drug resistance-related mutations in Candida auris. Machine learning has also been used to guide empiric therapy in Cambodia, which is a low-income country where invasive bacterial infections cause high rates of mortality. Researchers developed a model that used 35 independent variables as input, which included relevant clinical information, demographics, and information about an individual's living conditions. Based on this input, the model was able to predict the pathogen's gram stain with 71% accuracy and predict whether the pathogen was susceptible to gentamicin with 74% accuracy, ceftriaxone with 80% accuracy, ampicillin with 74% accuracy, or none of the above with 85% accuracy. Researchers noted that this model has the potential to improve targeting of empiric therapy, improve patient outcomes, and reduce the burden of antimicrobial resistance in low to middle income countries. Machine learning has also been used to classify groups of human microbiota, especially those that can be difficult to classify using traditional approaches. Researchers used 16S rRNA sequences as input to the model and tested it on 5 separate benchmarks. In the first benchmark, the model was able to predict whether a group of microorganisms was obtained from someone's external auditory canal, their gut, their hair, their nostril, their oral cavity, or their skin with 100% accuracy. In the second benchmark, the model was able to predict 12 unique locations on the skin that a group of microbes came from, including classes such as the volar forearm, the plantar foot, the forehead, the palm of your hand, etc. And it was shown to have 74% accuracy in this regard. In the third and fourth benchmark, the model was able to predict with 100% accuracy which individual of 3 a group of microbes was collected from, and in the fifth benchmark, the model was not only able to predict which individual a group of microorganisms came from, but whether it came from their left or right hand with 100% accuracy. Now, the final example I am going to show involves the use of a machine learning model to predict hemagglutinin and neuraminidase antigens of influenza to guide vaccine development. Researchers used previous hemagglutinin sequences along with hemagglutinin inhibition assay data as inputs to the model. They were able to achieve 96% accuracy in predicting the antigenicity of influenza in the period from 1997 to 2011, noting that their model significantly outperformed the World Health Organization's previous recommendations and could be used to improve the vaccine recommendation process. And here I've just listed some information that is useful as input to a machine learning model in the context of medicine. A lot of this data is readily available, and it is often used to train machine learning models that are currently being used in medicine. And if you're interested in reading more about any of this, here's a list of the references I used in this section of the presentation. So now that we know how machine learning generally works and how it has been applied in modern medicine, let us take a look at a problem that could be solved by machine learning in the future. Antimicrobial resistance is an emerging problem, with resistant organisms causing about 10 million deaths each year. One contribution to this problem is the lack of discovery of novel antimicrobial agents. The last class of antibiotics that was invented were lipopeptides, which were discovered in 1987. Since then, we have been in what is referred to as a discovery void, during which there have not been any novel antibiotics discovered. So here, I have put together a general outline of how machine learning models could be used to aid the development of new antibiotics. Now, each arrow in this diagram indicates a neural network, denoted NN, with the input to the left and output to the right. So we will start by looking at the first step, which involves use of a machine learning model that uses a whole genome sequence as an input and returns the structure of a protein as an output. In order to develop a new antimicrobial agent, we first need a way to identify potential targets, such as bacterial enzymes. To do this, we can sequence the genomes of different microbes, looking for sequences that differ from humans. Once we have a sequence that is significantly different from humans, we would want to predict the structure of a protein that corresponds to that genetic sequence. And in 2021, researchers at Google developed a machine learning model called AlphaFold, which, as I explained earlier, can do just this. Aside from predicting bacterial enzyme targets, we may also want to predict the structures of different peptides that can pass through bacterial cell membranes or that can insert into the membrane and create pores like lipopeptides do. Knowing the structure of proteins that do these things, we could use a machine learning model like AlphaFold to predict novel peptides that have similar shapes and similar properties to known peptides. Now that we have a protein structure, we're going to look at the next step, which involves the use of a machine learning model that takes a protein structure as input and predicts molecules that can bind to it and inhibit them, known as ligands. Now, once we have our predicted protein structure, we want to design a model that can do a couple of different things. For one, we want to predict whether a given molecule will bind to a given protein. And just this year, in 2022, a group of researchers developed a machine learning model known as DiffDock, which can accurately predict the binding location and orientation of a ligand on a protein. While this is useful, in the future, we would also want to develop a model that can predict the effect that such a ligand binding would actually have on the protein. We want to do this because to develop a new antimicrobial agent, we want to find a drug that can inhibit a bacterial enzyme. And ultimately, we would want a machine learning model that is capable of outputting the structure of a ligand that can bind to and inhibit a bacterial protein, which would be given as an input. And it should also be noted that you do not need machine learning to accomplish many of these computational tasks. Although machine learning can be very accurate, the main benefit is the speed at which it can solve problems. For example, I'm currently involved in research in which I'm trying to develop a machine learning model that can predict hydrodynamic properties of a protein, which basically describe how the protein moves in solution. And that's based on an input of a three-dimensional protein structure. Now, there's already a computational solution to this problem. However, it can take upwards of 5 to 15 minutes to get results for one protein. The machine learning model that I am working on is able to output the same results in about 0.02 seconds. So that means that in the same time the traditional computational methods take to analyze 1 to 3 proteins, the machine learning model can analyze about 45,000 proteins. And similarly, there are already computational methods to predict ligand binding to proteins, known as docking algorithms. However, a machine learning approach would be much faster, allowing researchers to get many more results. And this may not seem significant, but researchers that are interested in designing new drugs in this way must run the same analysis repeatedly with different ligands and different proteins until they find a potential drug candidate. And therefore, developing highly accurate machine learning solutions could vastly accelerate this process, potentially allowing us to discover new drugs much more rapidly than we ever could before. Now, if we develop a model that is capable of predicting molecules that can bind to and inhibit bacterial enzymes, we would also want to be able to predict off-target effects. And traditionally, animal and human trials are conducted to test these effects, but the development of a model that can predict effects ahead of time would be useful in reducing the suffering and death of animals and humans in this process. Now, one thing you may want to do is directly look for off-target effects on various proteins. You could use a similar model that was used before, and you would want the output to include something like the probability that your ligand will bind to and inhibit a wide range of human proteins. And this would allow you to predict whether your drug will have adverse consequences and may allow you to figure out how to mediate such an effect. And you could even use the exact same algorithm as before and just repeatedly run each protein with your ligand and get a probability that the protein will be inhibited. Now, you may also want to predict how your drug would interact with other drugs. As such, you could develop a model to predict the probability that your new molecule would interact with other drugs. The input in this case would be the structure of your ligand, and the output might include the probability that your drug will interact with another drug. And you could run this many times to get a table of probabilities of interactions between your drug and other common drugs that are prescribed. And you might even want to use the machine learning model to try and predict the effect that such a drug interaction would have. Now, instead of just predicting specific interactions, you may also want to predict general side effects. In the future, we might design a model that can take the ligand structure as an input and output something like the probabilities of different side effects. And maybe you'll get an output at like a table like this with each probability corresponding to a different side effect. So in this case, nausea, seizure, death. You may also want to develop a model that can predict the effect that a drug will have on different microorganisms. And you could design a model that predicts the effect of your ligand on a wide group of organisms, like the effect a drug would have on gram-positives, or you could design a model that predicts the effect on a specific species, like the effect it has on Chlamydia trichomonas. And maybe you would get a table at the end of it with probabilities of your drug inhibiting different species. Now, overall, machine learning technology is still relatively new, and many of the applications I've discussed in this part are unlikely to be figured out in the near future. However, like I said before, our machine learning models will only get better as time goes on. Now, in the far future, we may be able to design a machine learning model that is able to take input that describes a patient and outputs a drug or combination of drugs that would be perfectly able to treat them. And if this model proves reliable over time, it is possible that it could be used directly by patients who could simply provide the input required and get a prescription for the drug virtually. However, in my opinion, it is unlikely that this will ever be the case. While machine learning models may become very powerful, it will continue to be important that physicians are still involved throughout the process. For one, machine learning models are not currently 100% accurate, and the results will still need to be monitored and compared to other methods in the context of making a diagnosis. Furthermore, even if our models do become 100% accurate someday, we would still need physicians to interpret the results. For example, if we were to create a model that could design the perfect drug for treating a patient, it should still be monitored by a physician to prevent unnecessary prescriptions where, for example, the model might think the patient needs X drug, but the patient is perfectly healthy and doesn't need it at all. And even at the end of the day, if a model proves to be 100% reliable and never makes any mistakes, there will always continue to be a human component to medicine, which I just don't think can ever be replaced. And again, if you're interested in reading more about anything I've talked about, here's a list of the references I used in this final section of the presentation. And that is going to be it. If you have any questions, feel free to send me an email or let me know if there's something I should have included or something that didn't make any sense. Thank you, and bye.